what we see based on the fossil evidence and the geological evidence as well as the molecular fossils is that there was a massive kill-off and extinction of perhaps over 90% of marine organisms, many of which went extinct, quite a few went extinct. A lot of them died and weren't found in the early Triassic and maybe only established themselves again much later.
Today's speaker is Dr. Shane O'Reilly. He's from Ireland, which is pretty awesome. He holds a Bachelor of Science in Environmental Chemistry, and in 2013 he earned his PhD in Organic Geochemistry from Dublin City University.
So I'm going to be talking about molecular fossils. So probably what all of you think about, including myself, when you talk about fossils, you generally think of the science of paleontology. Classic paleontology involves, now I would admit this is a pretty gross misinterpretation of paleontology, so do any of you know the scene where this is taken from? You can probably guess, Jurassic Park. So in itself this is probably not overly scientific, but what I generally think of, what a lot of people think about when they think about fossil hunting and paleontology, they think about teams of scientists gathering around some rocky outcrop, slowly excavating, brushing away and digging away and revealing this spectacularly preserved skeletal remains of some really spectacular organism. Oftentimes we think about dinosaurs, they're really cool, of course we'll think about those first. But if we kind of take a step back and you actually think about how life and the Earth has developed, and Earth history and the origins of life, and even life today, how modern ecosystems are structured and how they develop, both on land and in the marine environment, we're talking about microscopic organisms. So organisms you cannot see with the naked eye, that are not large like the ones we've just been talking about, like dinosaurs, large organisms, complex organisms such as ourselves. We're talking about microscopic life. So the basis of most food webs and ecosystems are primary producers, and particularly in aquatic environments such as in the oceans, microalgae, microscopic algae, plankton, as well as cyanobacteria. These are the main players in terms of total mass and volume in ecosystems. And even when we look at the primary consumers, those organisms that chase after these guys and consume them, use the organic carbon that they produce by photosynthesis, we're talking about microscopic organisms as well. And then I kind of crudely have put in the rest, so the larger organisms. As you move up through the trophic levels, populations, abundance is much lower, and in terms of total mass, it's also much lower. So these are the types of organisms that we're actually talking about when we talk about life in a lot of cases, and that's kind of glossed over sometimes. And a lot of recent advances in terms of technology, looking at the genetic component of life in the environment, we're revealing a huge diversity of microscopic organisms, both eukaryote organisms as well as bacterial and archaeal organisms that are extremely diverse. And one other thing that's worth recognizing, and it kind of goes back to what we're talking about, if we look at Earth history from the present day down through the eons to around 4.6 billion years ago when the Earth formed, here is 540 million years ago. It's a long period of time, but in terms of the global or the total length of time over which Earth has been around, it's a very small amount of time. And this is where all of these large fossils are found, in the Phanerozoic. So this is where we have the radiation of large organisms with shells and skeletons. So what about life and the Earth history for the other 80%? We're talking about very small organisms. We're talking about microscopic organisms that did not produce large shells or skeletons that we think about when we think about fossil hunting. So we need some tools, various different tools and various different expertise to address these kinds of questions and learn about these periods of time and even ecosystems and life in this period where we do find large fossils. So myself, I'm an organic geochemist. You could call that a molecular fossil hunter of sorts. So we work closely with paleontologists and geologists. And what we do is the tools of the trade for us are pretty advanced modern analytical instruments that you find in organic chemistry labs. So we will get a rock and we actually powder the rock, we grind it up and we extract out the organic molecules that are trapped within those rocks. So this is just a glass vial with an organic extract in it. You'd probably think that looks like oil, crude oil. Well, it's pretty similar to what it actually is, and that's a good example of a really large deposit of molecular fossils. So oil and coal are really good examples of preserved molecular fossils and organic material. And what we do, so this is a really complex mixture of lots of different compounds, hundreds if not thousands of different organic molecules. So how can we actually go about analysing and interpreting this? What we do is we add a tiny amount of this onto what's called a gas chromatography column. And what that is a really thin tube, 60 meters long, but only about 0.25 millimeters in diameter. And we inject a small amount of it onto this and it passes through. And based on the different classes and the chemistry of the different compounds here, they come out the other end separated because each of them will interact differently with the internal walls of this tube. So you get this nice separation. So this is just time, essentially, and this is the amount of material. And we then blast this with electrons and look at the mass spectral fingerprint using mass spectrometry. We don't need to go into the details about how these work. Of course, we can talk about that in a little while if we want. But this is kind of the background, the practical background of how molecular fossil hunting or the field of organic geochemistry goes about its daily business, essentially. So I've been mentioning organic molecules and molecular fossils, and what I'm talking about and what you're right in thinking about biomolecules. These are the building blocks of life as we know it, and they're the basis or the currency of ecosystems. So the primary producers, they photosynthesize, they produce their own organic carbon to build their cells, and then the consumers use that organic matter for their own survival. So we talk about protein, you've heard about protein, carbohydrates, nucleic acids, so these include DNA and RNA, as well as fat or lipids. So these are the main types of biomolecules. But if we talk about molecular fossils, we are looking for particular types of organic molecules that will tell you something about the organism and can also be picked up and found in the geological record in rocks that are really, really old. You probably all know quite a bit about DNA and protein and the whole revolution in technology in terms of DNA sequencing and protein sequencing, and certainly there's a huge amount of information to be gained from these types of biomolecules. But the problem or the caveat there is that we actually don't find them in geological strata very often. You're typically talking about tens of thousands of years at best. These really pretty rapidly are degraded by both non-biological and biological processes, so we can't pick them up. So that leaves us with one of the main types of biomolecules which most organic geochemists think about are fats, essentially, so lipids. So based on their chemistry, they typically are long carbon-carbon molecules of long chains. Sometimes they can form cyclical compounds. Based on their different structures, different organisms can produce diagnostic molecules that will tell you something about the original organism. So bacteria produce certain types of lipids, eukaryotes produce certain types of lipids, algae, animals, they're included within the eukaryotes. They all produce certain types of lipids. So not as much information from DNA and protein, but the real powerful part of this is the fact that lipids can hang around for a very, very, very long amount of time. So certainly millions of years, even hundreds of millions of years. And we know that because good examples are coal and crude oil. So essentially this is kind of the principle of organic geochemistry and molecular fossil hunting. We have some organism, I won't tell you what it is because I'm not sure what it is. It's just a crude drawing of some type of swimming organism, some algae or bacteria. It's swimming around, it's doing its thing, it meets its demise somehow, it decays, and a portion of possibly the membrane, which is mostly lipid, can be preserved, will sink to the seafloor and be buried in the sediment, and over geological time this may hang around, as well as a lot of other lipid molecules. And then someone like me will come along and dig up this sediment that has become lithified to a sedimentary rock, and we can pull out these molecular fossils. So that's kind of essentially how it works. So that's kind of the background, the practical side of things. And for most of this talk, I wanted to bring you on a kind of a molecular tour of Earth history. So obviously we don't have that much time here, so we can't take a lot of stops, but we're going to take some, maybe perhaps biased on my part, interesting stops along the way. And the first stop is not going to be too far. It's going to be in terms of human history and archaeology, so how we can apply molecular fossils that we've been talking about to provide information to archaeologists and questions in archaeology and human history. So we've been, if you know a lot about the Neolithic times and the fact that around 9,000 to 8,000, perhaps even 7,000 years ago, depending on the location, we have sedentary lifestyles being established, agriculture being developed, and more complex lifestyles, cultures being developed. Oftentimes the remains, whether it be dwellings or remains of the early humans, it's pretty scant, there's not that much, but one of the main types of artifacts that you will find are pottery fragments, or they're called potsherds. They usually are pretty abundant, sometimes are the main type of artifact that you will find. And a really cool application of organic geochemistry and the use of molecular fossils is that we can go and take one of these potsherd fragments and we can scrape off a bit of the interior portion, and we can extract out the molecular fossils that are preserved within the ceramic itself. And sometimes certain lipid molecules would be diagnostic for example, cooking of fish, things like that. And what I've shown here is actually one example of, it's a cross plot here, so an X and a Y axis, and you've the two main fatty acids. So fatty acids you might have heard about in nutrition and diet. So these are these long chain hydrocarbons with this functional group here. We don't need to worry too much about that. But what you can do is you can identify them, and you can look at the isotope composition of these fatty acids from your particular sample. You can then look at the same fatty acids and the isotope composition of different products, whether it be meat or dairy products, and you can see where your samples plot. So they might be plot down here and would provide you with the evidence that it might have been a dairy product of some sort. A perhaps more, maybe kind of less pleasant or gruesome application of this would be in archaeological grave sites, or even much more recent in terms of forensics. One of the things that you find in a disrupted grave, possibly that it was only a temporary site for a body to be left behind, or maybe water would have washed away a lot of the material, but you can get this disgusting fatty material that gets left behind, and from the decay of the body tissues. And there's maybe a new approach where people are trying to look at the same types of fatty acids and isotope composition and see where it plots in terms of other fatty tissues. So if it plots up here, you know it was possibly human remains that was here. So it's of interest, particularly in forensics. It's and could be important in the future. So another I think nice application in terms of human history and archaeology is looking at fossil feces, fossil poo. It's technically called coprolite. And one really nice example is from a colleague who I work with in MIT. Her name is Inara Sistiaga, and she published a paper recently. She's an archaeologist who applies these kinds of tools in her own questions, and she has done a lot of work at some of these early Neanderthal sites in Spain. So these are 50,000 years old, and this essentially was a type of a cooking pit where these Neanderthals were hanging out, and what they found amongst this was some microscopic particles that they suspected were possibly feces within the cooking pit. I know it's a bit strange, but that's what was happening. But really they confirmed this by looking at the molecular fossil component of these small particles. And what they found were these kinds of complex lipid molecules here. So this is somewhat similar to cholesterol. So you know a little bit about cholesterol in terms of diet and nutrition, and you know about plant sterols. Well, these are similar kinds of compounds, but there's some structural changes here that only occur as your food passes through your intestine, and the intestinal bacteria will make certain changes where they'll concentrate these types of fecal sterols in your feces. And they found these types of molecules, so they confirmed that they were, in fact, it was poo. But also the more interesting thing was they found plant-derived fecal sterols, so sterols with some of these side chains that are from plants. And that was nice evidence that in this site and in many other sites possibly that Neanderthals had quite a mixed diet. So they weren't just meat eaters, they were actually consuming vegetables as well. So that's I think a really cool application. And OReilly is kind of moving ahead with this and trying to apply this further and develop this type of approach to look at paleo diet in some of these early archaeological sites. If we stay with the poop theme, and we look now, actually we're going to go, I guess, more recent in time, but a recent paper that's been published is the culmination of a lot of work which I've been involved in previously during my PhD in Ireland, has been spearheaded by Bill Mahaney in the University of York in Canada, and he's had a passion for this for quite a number of years. But it's essentially Hannibal's route over the Alps. So Hannibal was a Carthaginian general. It was involved in the Punic Wars, one of a fantastic story in human history and human warfare. And one of the age-old questions is, which route did he take over the Alps into modern-day Italy? And based on Bill's combing of the historical literature, as well as combing the Cottian Alps and the various different routes, he came up with the hypothesis that he thinks the most likely route was probably the Col de la Traversette, this route here, based on the morphology of the rocks and how certain historians close to 218 BC, close to when this actually would have occurred itself, described the route that these would have taken. So obviously we want to get some better evidence and some more evidence that this might have been the route. And he described, he found a two-tier rockfall that was described in some of the historical texts. And close to this two-tier rockfall that he found on this route, that may or may not be associated with this historical text, of course, there was this large flat expanse that was completely stood out in terms of the other types of terrain, that was much more rugged, and it would have been a perfect watering hole campsite. So he came up with the hypothesis that this maybe was an ideal campground for his army. Now this would have been thousands of soldiers, thousands of horses, and a certain number of elephants. So there should have been some kind of evidence left behind of some types of activity. So we dug some soil pits and we described the soil horizons, and at around 40 or 50 centimeters or so down, there was a real disturbance in the soil horizons, which is interesting in itself. But when we dated it, it came back using radiocarbon dating. It came back as around 218 BC. So we then went and used some of the tools that I've been talking about, as well as looking at the bacterial composition of these different soil layers, and we found fecal-derived deposits. So this churned up layer looks like it may have been a mass animal dumpage of sorts. Now, whether it was other types of animals, we're not sure. It's tantalizing evidence that this could have been some site at that particular time where there was an awful lot of animals passing through. And of course, it's not a smoking gun by any means, but it really is an interesting story. And I think what the clincher would be to obviously do some archaeological studies to try and confirm this, try and find some equipment, some coins or something like that. So if we go away from human history now and we go a further step back in time, we can go 252 million years ago to a period of time at the end of the Permian, which is often called the Great Dying. And this is one of the largest mass extinction events known to have occurred on Earth. So what we see based on the fossil evidence and the geological evidence, as well as the molecular fossils, is that there was a massive kill-off and extinction of perhaps over 90% of marine organisms, many of which went extinct. Quite a few went extinct. A lot of them died and weren't found in the early Triassic, and maybe only established themselves again much later. We also find the increased evidence for a lot of what are called disaster taxa. So these include a lot of microbial type ecosystems, microbe dominated ecosystems, such as we find in the Precambrian, which we'll talk a little bit about. So these weren't around that much in the Phanerozoic, but they popped up again after the extinction event. And that kind of lines up with the fact that a lot of these large complex organisms that graze the bottom and would remove a lot of these microbial mats that would have been present, they died off, so you saw these microbial mats and microbialites being established again. Now there's this is a very active area of research, and a lot of different theories about why this happened. One certainly that probably had a major part to play was the massive volcanism that's been found at this time, that's been Sam Bowring in MIT has studied, and this probably had a catastrophic effect on life at this time. There's also some other theories about global warming and massive release of methane from the seabed that would have caused something like a lack of oxygen in the ocean, sulfide, which is really toxic to us and most complex organisms, and this would have maybe been some of the causes of this extinction event. So I've mentioned briefly about microbe-dominated ecosystems, and one which I've studied during my PhD these are called methane seeps. So with advances in technology, we can map the seafloor. We have mapped the seafloor to a really good degree, and we're mapping more and more of the seafloor each year. And what we found is there's a huge abundance of these methane seep ecosystems. So these are ecosystems on the seafloor, predominantly in deep sea settings. And what's occurring here is that you have methane that's seeping up through the seabed through the sediment from depth to the seafloor, and specific extreme groups of archaea and bacteria are utilizing this methane. And one of the byproducts of this methane is calcium carbonate or limestone. So here's a microscopic image of a quartz grain being coated by calcium carbonate crystals, but what's really cool is that you get very large macroscopic dramatic structures on the seafloor. So these are calcium carbonate slabs. So this is an example from the Irish Sea. These actually form huge mounds, tens of meters high, and these types of carbonates in these methane seep ecosystems can be hundreds of meters across. And these are really important in regulating and controlling the release of methane to the water column and the atmosphere. And we know methane is an important greenhouse gas. We've mentioned it just previously in terms of its potential role in mass extinction events. And so this is one cool example. But why I kind of bring it up is that we can find these types of carbonates in extremely old rocks. So we look at marine carbonates. This is an example of 300-million-year-old marine carbonate, and we find the same types of lipid molecules that are found in the modern ones. So these, we don't need to look at the details here, but these are all different, very unusual lipids that only certain types of bacteria and archaea produce. And we can also look at the isotope fingerprint of these, and we know they're using the methane. So we're demonstrating that these were around at that time, and that this type of ecosystem was established at 300 million years ago, and probably much older, and we might find more evidence of that. So if we move further and further back now, we're leaving the Phanerozoic, we're leaving where most of the fossils are found, and we're going into the Precambrian. So from 540 million years and older, we're talking about Precambrian Earth. And the unfortunate thing is that a lot of, due to plate tectonics and weathering, we lose a lot of the rocks from these times, from this early Earth, which is unfortunate, but there's some very interesting questions and some really interesting areas of research that are ongoing. And one of them, of course, is from the Cambrian we find there's a massive radiation of animals and complex life. But when did that occur? What were the foundations of complex life? Is a really interesting question. Now, Roger Summons in MIT, who I worked with in the 90s with some colleagues, found in some well-preserved Proterozoic rocks very unusual molecular fossils. Again, these are kind of similar to the cholesterol type molecules that were abundant in these rocks, and they have very unusual structural changes. And what he proposed then was that these were from early types of animals or sea sponges. So the only organisms, or one of the only organisms that produces these types of lipids are sea sponges. But the kind of more conclusive evidence came just in the last probably month or so when a colleague in the lab, David Gold, published a paper where he used molecular clocks and biochemical pathways of these various organisms that we now have genomes for. And he combined that with the molecular fossil approach to confirm to a good degree of accuracy that indeed the sea sponges probably were around at that time, and they probably were the organisms responsible for these molecular fossils. So that's really tantalizing evidence that indeed these were there, and these may have been among the earliest animals on the scene. Again, as we move further and further back, we're getting really into a kind of murky, deep past, and it's very difficult to study these types of environments because we have only a handful of rocks that are present, and a lot of them are so altered by metamorphic processes that they don't preserve maybe a lot of the geological isotope or molecular fossil information that we would love to be able to find in these types of rocks. But this is when life essentially evolved. This is, where the organisms that were at the really early stages of Earth history, they came about, they developed. And we have some really nice examples in a couple of instances of the earliest evidence of life. So these are deposits of rock in Australia. And what these are layered microbial mats that have been lithified and that grow with time through the action of certain types of bacteria. And this is an example of a modern microbial mat. So it's slimy, sticky, and it has some kind of layering and structure. And how do we know that? We know that because remarkably, a lot of these types of microbial structures are found in very restricted number of settings today on Earth. So we have in the Bahamas and Western Australia, in Shark Bay, we have what are called stromatolites, which look like this when you look at them when you section them. And we can go and look at the organisms that are present, that are being involved in building these modern examples. And this will tell us an awful lot about the older examples. So we use them as analogues to understand this deep past. And of course, naturally, you'll probably be thinking about the hunt for evidence of life elsewhere. And we have the Curiosity rover, which is currently trucking around on Mars, taking samples. And it has some of the kinds of capabilities that I've just been talking about. It can look at the molecular constituents, the organic component of rock samples that it takes, of soil samples that it takes. And the hope is that it may find evidence probably of more likely microbial life on early Mars rather than the current evidence for life. Perhaps future missions might say something more about that. Perhaps we can get some samples back. So I hope to have maybe just sparked off or started a discussion that we might continue. I'll just quickly thank some of my collaborators and some of the funding bodies and collaborating institutions. And I will certainly thank you all for listening. And if you have any questions, absolutely just go ahead. Thank you.
I'm very interested in bog ore and the bacteria that creates bog ore. Now, is it true that a lot of iron deposits, ancient iron deposits like the ones in Western Australia, are the result of bog ore, or how were they developed? How did they accumulate there?
So you're talking about bog
ore. Bog ore is very well known around here. It was the basis of the colonial iron industry.
Yes.
And bog ore accumulated on the rocks, and they're found in a lot of the ponds and the swamps.
Yeah, that's typically deposits like that, minerals like that, are often formed from microbial reduction of iron in conditions without oxygen. So certainly, I don't know too much about those particular examples, but certainly in terms of the microbial and microbiological role, I think it's probably iron reducing bacteria that would be involved in that. So yeah.
How far back does that go to?
Well, that particular metabolic capacity and those types of bacteria are essentially, we're talking about really early Earth, because as we know it, the life for much of the early Precambrian Earth would have been anoxic, so there would have been no oxygen. So it would have been those types of anaerobic bacteria that would have thrived. And then with the evolution of cyanobacteria, which can photosynthesize, which were almost certainly the first photosynthesizing bacteria, they produced oxygen, and they changed the scene completely. So. In terms of fossil preservation generally, but certainly for molecular fossils and organic preservation, the best examples are found when you have rapid burial and the removal from oxygen and oxic conditions. So that's certainly one of the best ways where you can get good preservation. Of course, if you have some organic remains or fossil remains and it just sits on the surface, number one, there's going to be a lot of organisms that can and will completely degrade everything, but the oxygen that's there, it provides the necessary oxygen for microbial processes, which are really, really efficient. So aerobic respiration, which is what we do, a lot of the bacteria that are around today also use oxygen and can really, really efficiently degrade and use that organic matter. So certainly that's key, that's critical, is to burial in anoxic settings, anaerobic environments.
What you discover is informed by anaerobic bacteria.
In certain examples here, or so a lot of the time you can certainly see there's certain types of minerals, certain types of rocks are only deposited in anaerobic settings. So one very good example, a modern example, is the Black Sea itself. So actually the water itself has no oxygen, and certainly the sediments have no oxygen. Usually if you go into a sedimentary environment, if you take a sediment core, it's the first couple of millimeters have some oxygen, and then it's totally depleted, and then it's these anaerobic organisms, bacteria and archaea that are present. So we kind of almost want anaerobic conditions, and yeah, so I guess it's certainly one of the preservation processes, and it maybe in some examples could complicate some of the stories depending on what you're thinking about. For example, the soil pit in the Hannibal story, there may be some types of bacteria that could produce some of these signals that we're seeing. So yeah.
So the molecules you find, the sterols and other molecules you find in really old rocks, how do you know that the molecule hasn't changed in some way?
Yes, so based on the biosynthesis of a lot of these molecules and how we study, if we study using degradation experiments or using natural settings, we find that there is changes to the molecules themselves. These lipids will change with time, but they'll change in such a way that it's predictable and that we now are getting a much better understanding for. So a lot of what's preserved in these really old rocks, what's preserved are the hydrocarbon skeletons. So you lose a lot of the unsaturation points and different function groups. They're all removed, but luckily the hydrocarbon skeleton is extremely stable. Most types of organisms, microorganisms, don't have the capacity to break up these strong hydrocarbon skeletons. So that's a lot of time that's what we're looking at. So there is a pathway from the biomolecule, the biolipid, to the geolipid, what's often called a geolipid. So that's a very good point as well. But that could tell you a lot about the type of environment that particular lipid was deposited. So there's certain changes to the lipid that occurs in settings without oxygen versus settings with oxygen. So there can be a lot of, it's not just the organism itself and the source organism, you can tell a lot about the type of environment as well. So yeah, but it's a very good point, yeah.
Would a consequence of your most recent comment be that a new type of dating mechanism?
It's usually not so much. So these are all organic molecules. So radiocarbon dating, of course, is already conducted and can. We're starting to develop methods to radiocarbon date individual compounds using these kinds of approaches. But radiocarbon dating only goes back so far. So the half-life of carbon-14 kind of really just limits the use of radiocarbon to tens of thousands of years. But there's other types of radioactive elements that people use to date really, really old rocks. So uranium series dating and some of these heavier elements are used to date ash layers, and the relationship between those ash layers and fossils and some of the rocks that we'll be talking about. That's how we get the kind of timeline right. So yeah, certainly this approach wouldn't be, and these molecules wouldn't be directly used for dating these types of old rocks.
You have these organic molecular fossils. Is there more information that you can take from them after you get them into the lab or something, aside from there was life and where possibly it was produced, or from what it?
Yeah. So some of the information that's contained within these. So you've got obviously, you can relate this particular type of molecule to this particular type of organism. Then you can look at the relative amounts of these different lipids to say, oh, these types of algae were more abundant than these types of algae or these bacteria. You can also look, as I've kind of mentioned, at the isotope composition. So the isotopes, we look at the ratio of carbon-13 isotope to carbon-12 isotopes of these individual molecules. And that can tell you about the biochemical pathway and the types of physiology that these organisms have been conducting. So a really nice example is the methane oxidation and these methane seeps. So they have a really fingerprint characteristic isotope depletion that is only associated with methane oxidation. So there's a whole layer of information that is there. And then I've mentioned as well about some of the types of changes that happens, these molecular fossils, only under certain conditions, under certain what's called diagenesis. So diagenesis is any change to a rock, sediment or organic material since it's from when it's deposited. And if you have oxygen present, certain reactions will occur to change the lipid in a certain way. Maybe it'll completely degrade it. Whereas if it's anaerobic without oxygen, you'll have different types of changes. So you can start to build a picture about of the broader environmental context and depositional conditions. Yeah, so they're the kind of main layers of information.
I actually have a question about the methane seeps. You said that one of the possible explanations for the Great Dying was that there could have been this change in the oceans from a sudden increase in the amount of methane. Has there been any effort, or do you think will there be any effort to try and use this approach to track, if you can track when and where there were these methane-loving collections of bacterial life or microbial life, do you think that you could potentially prove that there was an increase in the methane and thus, lend credibility to that explanation? Yeah, I mean, that's very much a very active area of research. It's something which I'm not directly involved in, but I know there's been some recent papers over the last 2 or 3 years that have proposed that these types of organisms were critical, and through maybe plate tectonics or some other changes in the Earth, there was a massive release of methane
from methane hydrates. These are gas hydrates you might have heard of. So these are methane molecules that are under certain depth and pressure conditions where they actually form these solid hydrates that are surrounded by these cages of water molecules essentially. And these are you know, present and substantial even today. So if anything changed the stability of these gas hydrates and released them, it certainly would have the potential to cause some pretty crazy changes to the water chemistry and atmosphere. So methane itself is very much a very strong greenhouse gas, much more so than CO2. So yeah, it is a really kind of hot topic. A lot of people are actively involved in this, both from geological point of view, but also looking at the microorganisms that may have been involved. So I think it's going to be, you'll certainly find some pretty interesting developments over the next few years, I think. So, yeah.
These methane deposits, are they created by bacteria or microbes?
Yes, yeah.
Underwater and under the...
Yeah, typically on the seafloor, you have the methane is seeping up, and the specific microbes are certain types of archaea and bacteria that reduce sulfate, so sulfate reducing bacteria. So these kind of work together to consume the methane, and pretty much just for their own survival. But one of the byproducts of that is calcium carbonate. So it's just purely a byproduct, and then you get these over large areas and timescales. You get these huge deposits, really dramatic. And other types of organisms then can use these hard surfaces as a substrate with which to develop and then the ecosystem flourishes. So it's based on.
So the limestone I'm familiar with, like the Dover Cliffs and the
Yeah.
district in England, those were deposited by microbes with a calcium skeleton.
Yes, yeah.
You're talking about a different type of limestone.
Yes, it's more of it's a passive almost product of their metabolism, and it's a byproduct. Whereas those organisms are skeletal or skeletal organisms. They actively produce skeletons, and yes, the Cliffs of Dover are really good examples of huge deposits of these microscopic algae, and they're specifically their skeletal remains. So.
And the limestone you're studying is much different. I guess,
Yeah,
and
it's
often older.
Yes, it can be older, and it's a different type of limestone. Yes, so calcium carbonate there's typically you divide it up into the skeletal type or the non-skeletal type, of which there is one of them is the kind of bacterial or microbial produced carbonate. So these stromatolites are types of are formed from calcium carbonate, and these precipitate by the action of cyanobacteria and probably other types of bacteria as well. So yeah, there's some.
There's cyanobacteria.
Yeah, cyanobacteria. So these were probably the first organisms on the scene that performed photosynthesis.
And these are anaerobic bacteria.
These are pretty versatile in what they can do and how they can exist, but they typically will be present in shallow, pretty much oxic waters, and they'll utilize the energy from the sun to do photosynthesis. So, but they can live. They're pretty versatile. They can live within rocks. They can bore into rocks. So a lot of them are endolithic. They live within rocks. So. And they can withstand quite a lot. They usually live on the border between very low oxygen conditions, maybe, and oxygen-rich conditions. So
some of the natural gases that's far underground is not produced by plants or animals, but actually produced by these ancient bacteria that live in the rocks themselves. Not,
yeah.
They think that's true.
Yes. So a lot of methane and natural gas that we use ourselves as a resource, it typically can be divided up into like biogenic or biologically produced, or thermogenic. Thermogenic is formed from deposits of organic rich layers, usually over a kilometre deep in the seabed, and the temperature increases to such a degree that it causes cracking of organic matter and the release of methane. But there's these organisms that we're talking about. There's also methanogenic organisms, methanogenic archaea, that live perhaps not as deep, in the hundreds of meters in the sediment in the seafloor, and they produce methane. So they are a major player in terms of the natural gas deposits, and they can, you know, be involved in forming a lot of these pretty diverse features on the seafloor. So yeah, so there's
So if you were given a sample of natural gas, can you tell what type of gas it was produced by?
That's been a really active area of research for quite a while in the petroleum industry and in the natural gas industry. And yes, you can essentially, based on looking at the isotope composition of the methane itself and some of the other small hydrocarbons, you can tell whether it was likely biogenic, likely thermogenic. And also then within the context of the seismic mapping, you can see where there are faults, which would be more likely then it would be more likely to have been thermogenic, so there's a much deeper source. Whereas if it's in the close in the tens of meters to hundreds of meters, you can sometimes look at the seismic evidence for gas accumulations and gas pockets within the seabed. So based on the chemistry of your gas and the geological context and the geophysical data, you can often come to that conclusion, where it came from and who produced it, I guess.
Would it be potentially possible to isolate these bacteria and culture them in laboratory conditions to produce methane as an energy source?
Yes, so a lot of microbiologists would be really very, very interested in that, and that would be of huge interest in terms of the biotech industry. These organisms typically are notoriously difficult to culture because they exist in such extreme conditions. It stands to reason that if you go and try and sample them, bring them back to the lab and grow them, it doesn't work because you don't have the conditions right to grow them. So that's why you oftentimes need to do culture-independent approaches, such as what we're talking about with the molecular fossils and the lipids, but also looking at the genes. So we can actually go and take an environmental sample and extract the DNA from these organisms, and then you can start to learn something about their biology and where they lie in relation to other organisms. But yeah, that would be fantastic, and I'm sure if there is a major advance in terms of culturing these organisms, that would have a knock-on effect in a lot of different disciplines, both biology, geology, and chemistry. So that would be a major advance.
So these microbes like high temperature or high pressure?
A lot of them, yeah, a lot of them like high pressure, high temperature. A lot of them like to be in sulfidic conditions. A lot of them hate any amount of oxygen, so they won't survive if you introduce them to a small amount of oxygen. It might even be much lower than the levels that are present in the lab or here, and that would be enough to kill them. And a lot of them, we simply don't know how they make a living. So of course we can't grow them then if we don't know what they're even doing. So that's it's a fundamental problem, but it's going to be interesting to see how we tackle that one. Thank you very much. Thank you for listening.
Sorry, Comcast is really insisting.
No, it's fine. Thank you.
We're constantly applying general world knowledge using our general reasoning ability to understand language from the bottom up, from all the way from sounds all the way up to meaning.